And the presentation will be divided into small parts. I'm going to give you a brief introduction about the history and how we at the end reach to have the molecular viewer that we are using. And then I'm going to focus on the 3D protein imaging, and in particular on the protein viewer that we develop. So how does our story begin? Our story begins with the crystallography. Actually, without the X-ray, the molecular viewing is not possible. So our history began in 1895 with the discovery of X-ray, and goes on thanks to Henry Bragg and Lawrence Bragg, that are father and son. And as you already know, probably they are the ones that determine the atomic structure of the crystal. So they are the first that using X-ray diffraction can actually solve a crystal, and in particular of the sodium chloride. And during the years, the X-ray diffraction actually helps a lot of scientists to solve many problems. And among them, I like to talk about women in science, as I'm one of them. And so I'm going to talk about Kathleen Nosday, that is the one that at the end demonstrated the Kekulé hypothesis, and so that the benzene has 6 equal size bonds and not an alternating double and single bond. So it's like one of big discovery. And the other woman in science that I really want to thank is Dorothy Crawford Hodgkin. That is the first one that solved the structure of penicillin and vitamin B12. And for this discovery, for this structure, she obtained the Nobel Prize in Science. So we hope that this discovery or this the importance of women in science can go on. And this is first. This is before the solving or something about the protein. The first protein was solved in structure was solved at mid 50s of the 18th centuries, and Kendrew and co-worker was trying to define how the structure was. They were trying to use the X-ray diffraction images and trying to imagine how was the structure in the 3D. So they just used this plastilin and Booth's model to see the myoglobin. So this is the first protein, and this is the first model that we have in history. So I really like it, and as you can see, it's 5 centimeters per Armstrong. And actually, it was really difficult to take this model to a congress or to show it to another scientist. So we need to wait until 1970 that Byron Bender invented a machine that can bend wire in following the backbone of the protein. In this time, there were already computer program for modeling protein, but you need a lot of money to have it into your university, and it was really expensive to create a model. And using actually the Byron Bender model, where really there is like a factor really that is really interesting and funny, because thanks to Byron Bender's model in a congress, 2 scientists discovered the superfamily of the globulins, because 2 different scientists with 2 different completely different protein, because they had only the 9% of sequencing that is similar between the 2 protein as the same folding. So it's the first time that we can see actually a superfamily. So it's really thinking about the time is really funny that using a wire you can see the protein. And then starting from the 1970 and going on, the computer programs and the computer power was increased. So using a computer, it was easier in the lab. And so actually the physical model, physical in sense of material model, just going from something that was useful to visualize the protein to something that is art. So Julian Vos Henry prepared this, that is a structure of a protein actually, and it's art. It was in a gallery. And now if you want to take the maximum notes in your biochemical or biochemistry exam, you can like give this gift to your professor, and it's not so expensive. It's glass, and with the laser they fracture the glass in order to create the structure of the protein. It's really something that you can give to your professor, or to, I mean, I think I'm going to give this to Gianluca, one of these, with a protein, with the amino acid oxidase that is his favorite protein. So going from to computational representation, the first one, the first time that we can see like in a computer or something similar to a computer, protein structure was the 1964 in the MIT in USA. And with an oscilloscope, they were able to visualize the structure of the protein starting from the X-ray diffraction pattern. So this is the first image of the computational image of a protein. And then starting from that, David and Jane Richardson developed the first computer program in North Carolina, in the University of North Carolina, and then they developed the first molecular viewer, as we know it, that is MAGE. And this is actually the first program. They were using it in their lab, and then extended for in the classes. And in one of these classes, the undergraduate student, an undergraduate student decided to implement the algorithm, and is the first one actually. So undergraduate student, so he didn't know almost nothing about anything, because computer science, it was still developing. He developed the small visualization, and this is the first time that we can see spheres for the atoms, sticks, that is this visualization, and you can rotate the image. Is the first actually viewer that as we know it. And from then to now, these are all the visualization that are the viewers that are available now. You have standalone application or web-based application, but and all of them have something that is good, but is something that didn't really work. I mean, you can visualize protein maybe, but you cannot have an image of high quality, or you can have an image of high quality, but you cannot really visualize it before you obtain the image. So it's really tricky, or you need some particular skills. So starting from this, Gianluca, that is the founder of the 3D protein imaging webpage, decided to develop its own viewer. He studied with me at the University of Insubria, where he took the degree master also in industrial and molecular biotechnology. But he has clear in his mind that the bench is not his life. So he moved from the bench to the computer. And while he was working there in the biochemistry lab of the Professor Poligioni, all the other guys or the other students were asking him to have a protein image to put in the thesis or in a presentation or in a poster. So he said, okay, we have PyMol in all the computer of the lab. Why don't you use PyMol? It's easy. It's a viewer commonly used in the lab. And they said that it's not so easy to use. You need to know the code. You need to know something. It was easier for them to ask to Gianluca because it was faster. It was like developing something to work with. So at the end, he said, okay, maybe I have to develop something that they can really use for free. And so he started this adventure. And only 1 year ago, Martina joined us in this project. Martina studied in the University of Padova, and she's like working in the part of crystallography actually. And now she's a postdoctoral researcher in the Scripps Research Institute in California, and she's not dealing with the scientific part of the project, but she's working with us for the social media. So she's the one that in the social media posts and give us some feedback about what we have to change or what the people likes. But why you should choose 3D protein imaging? Well, it's an online tool, so you don't have to download anything. You don't have to install nothing. It's really, really easy to use for beginners because you don't need to script at all. You just have to do a click with your mouse. At the end, you can obtain a high quality rendering that is not so common because most of the program available now give you back low quality images or doesn't allow you to have a rendering at all. And what I think a feature we are really proud of is the fact that you can share the work and the project with your collaborators. So what you can do is just prepare your project, your structure, your protein with the color you need or the representation you've chosen. You can send it to your boss. He can open the project in the 3D protein imaging and say, okay, I don't like this color. He can change the color himself without sending an image, receiving an email when he said, I don't like the color, I don't like the orientation, you should choose another one. You can actually work on the project alone. So how does it work? You load your protein, you change wherever you want, so representation, color, and orientation, and in a few minutes, you will have a final high quality rendering, so a final high quality image. How does it work? This is the interface of the protein imager. So you will see this screen. Oh, sorry, and you can load almost a lot all the structure available. So you can load a ChemDraw file, you can load a PDB, so protein data bank file, international crystallography, and everything. If you have your project on your computer, your structure on your computer, because you have solved it, you can also load it by just drag and drop in the figure. Another thing that we can give you, it's a pre-style. So you can have a default one or a good cell or cell paint style. What does it mean? The default style is the classical style that you can see in the PyMol when you load the structure. The good cell-like is the one that is similar. This is the default. And this one is the good cell-like, is for the ones that are familiar with the PDB data bank. This is the molecule of the month style. You can see this very often in the PDB. And also you have now the cell paint, that is another software that is available online that has been developed to see cells. So this is more like used to have like more simplified version of your protein or your biomolecule. Another thing that you can do if you are not familiar with the 3D protein imaging is that you can load example project so you can see how to perform, how to reach a certain result. So you can load a predefined project and you can see how that effect was obtained. So when you load the protein, what do you see? This is the interface. You have here the viewer itself, in which every change you made you can see like almost directly. You have a right panel in which is shown the structure and the part of the structure. A right panel in which you can actually modify the selection and a bottom panel that is for advanced features. More in detail, in the right panel structure, you have the first part, gives you the possibility to change between two different structures, because you actually can load more than one structure, and then you can compare them by aligning the structure. You have the information about the structure that you are visualizing. So you will have the code. This is for example, a PDB code, and the description of the protein. And the last part here is about the selection. From here you can change everything, representation and color of your selection. Here you have an example. You can have all for the protein. You can have all these representation and spheres, surface, tubes, sticks, and cartoons. And one other option that is good to use if you have small selection is that you can label the residue that you have in your selection. It's really easy to use. You just have to click on it and you will see the change. And you can also change all the colors. So you can have this style that is the good cell coloring. Then you can have the uniform one, so just one color. You can color by element, so each element will have a defined color. You can have in gray the carbon, in blue the oxygen, and go on. Or you can color by moiety. In this case, I've colored the moiety is the beta sheet or alpha helix rendering, and this is the beta sheet, this is the alpha helix and the random coil. But you can change, you can choose to color by residue, by chain. You can actually do a lot of things. Then you can color by proximity. In this case is proximity from the ligand. This is the ligand here, and from the ligand to the less I don't know how to say that in English, neither in Spanish. And like here is the most it's near the substrate, and here is really well, you understand, because now I cannot say that in English. Okay, and then you can color by bifactor. Sorry, here. Okay, this is the bifactor that give you the information about the mobility of a residue that is strictly correlated to the temperature. Okay, then you have this panel that is about the selection. Actually, you can select everything just using a click. So if you want to select a residue, you can just click on the residue. You want to select the chain, you just have to click on the chain. And all you can do this by using enable here chain, models, or residue. You can select or deselect actually from the structure hierarchy. You can erase part of the selection by removing a chain or removing a residue, because you don't want to see that in your image. Or you can use this advanced selection to select by entity, proximity, range, or property. Here you have an example. This is the horseradish peroxidase, an enzyme that we don't really know. And I have selected the lysine and the arginine on the surface. So it's really easy. You write here what you want to select, in which selection you have to look for that residues, and you just create the selection. You just click here, and on you will have a new selection. You can change everything of that selection. Here I just show the lysine and the arginine on the surface in red, and it took me like 2 seconds. While in PyMol, I've tried, and it's like you have to remember how to write the code, and you put a space. Is not, you shouldn't have put it, so the selection is with anything inside. So it's like, for me, it's complicated. Here is more easier like this. And then you have the last panel, that is the bottom panel. You actually have a lot of features here. The first one is the log history, just give you information on what you have done with that structure up to that moment. Then you have the sequence viewer, that is like the one that you have in PyMol, but for me it's more easy to read, because actually you have the name of the chain, the name of the protein also here. So, and also from here you can select the specific residue on the viewer. You can create distances, you can change every color, you can change the dimension of the label, you can change the connector. And this is a new feature of the protein imaging, and you can actually visualize membrane. You can visualize membrane like this one, or you can visualize membrane like this one. So you can change color, you can change the spacing, you can change the dimension of the membrane, and only by modifying these parameters. And this is the most complex part of this panel, because you have different preset styles also in this case, and these settings will change according to the preset style. I have a video, because I know that just talking about and I think is like boring, so I've just put a video so you can really see how does it work. And here, so this is always the oxidase peroxidase. Okay, you can see that you can really move the protein as you wish. And from this part of the viewing option, in the preset good cell, you have these things. You can actually change the outline color. So the outlines are these lines that define the spheres in this case. You can change the color. It is immediate. You just move the mouse and they changes. You can put the grayscale. You can change the background, and it's immediate. And actually you can see in the same time that you change the color, you can see the changes. And you can add the shadowing, or you can remove it. And if you change the pre-style, you change all the settings. This is the real one, preset style. You can change the type of view, you can change the outlines also in this case, you can add it, you can change the color, and you can do a really lot of things. You can clip the plane, so you can cut the part of the protein and see just what is above the plane. And you can add these features like to see the 3D structure in a more defined way. You can see what is in front of you from what is most far away from you. And okay, you can change everything, almost everything. And this is something that gives you the idea of the 3D structure of the occlusion, or where is the residue, is like near the surface or more is depend on the structure. And you can see everything here, and it's really easy actually. And this is the final rendering. You can see that is practically identical. It's identical to the viewer, and it takes like 5 minutes to obtain an image that you can use in a thesis, in a paper, because it has 300 dpi of quality, and I've used as background in a poster, and it doesn't lose quality. And then there is the last panel. This is the part of the rendering options, and this panel gives you the setting of a part that is not linked to the viewer, but to the server-side rendering of this program. So you can change the transparency of the objects, and you can choose the cartoon and the nucleotide type. And also, this is about the ambient occlusion. You see here, this is the default mode, the inverted one, and it is disabled. You won't see any difference here because the image is not so good, but I have another video for you because I like to take video. And this is the protein that I render. So I went to the rendering option. This is what I've shown you. This is the default mode. You can see the transparency is almost inexistent. But here you can see that the surface is like in the same plane of the, or under the structure, and here is disabled, so you don't have any differences between the two planes. Okay, you can change the type of background. It could be transparent or opaque. You can choose to have a monolayer, so you won't have any differences between the different planes in the transparency, or you can have different layers. Here are some other options about the transparency. Sorry. Here. Okay, and here, this is my favorite part. You can change the representation of the cartoons. My favorite one is the cylindrical helices. I really like the shape of the cylinder, but you can also smooth the cartoon by putting the oval representation, or you can increase the angles by using the rectangle one. You can change also the nucleotide type, and you can choose to have the classical shape, or you can put the ring, or you can really mix a lot of things here. And sorry, this is another video. I have a lot. This is a rendering from the starting. It's a more complex structure. This is a ribosome, so you have protein, DNA, and lipids. And you can see that the viewer can handle structure with a lot of atoms, and it's really easy to work with. And here you will see actually everything you can do with this viewer. So we select the protein selection, and we change what we see. So we want to erase a chain. We just took a click, and from here it disappeared. Then we create a new selection. We name it as we wish, and we decided to add the same chain that we have erased from the other side. So why to do that? We want to change the color. We want to highlight that chain is it's a lipid, and we want to change the color, just to say, okay, it's not part of the protein. And I think that this in PyMol took a code or a line of a code that it's impossible for me to remember. So you have changed the color, you have edited everything, and you just resize the image as you wish, and it takes 1 second. You can change everything, the deepness of the shadowing, and with a click, you just get image, and in 3 minutes, you have the rendering. You just click, you have a notification, and you can then download the image in a really, really high quality. It's really easy to use, as you have seen. and you can really use it during a meeting. I use it very often while we are working with the FET Open outside. That is it's a project in which we want to we want to create a cascade, an enzymatic cascade, because we want to create a system that can compete with the actual one that is using the chemical a chemical reaction. So what we want to do is to have in the same pot 2 different enzymes that has different temperature optimum, and that can have some cofactor or product that are sensible to temperature. So what we want to do is to immobilize on magnetic nanoparticles, selectively activate the enzymes that are binding to this particle. Why? So this is my part of the project, and what we are doing now is we have the particles with carboxylic group, and we want to functionalize them with these NTA copper complex that will chelate the histidine tag that are present on the enzymes. To do that, we need to know the structure of this enzyme. We need to know if there are some other histidines that can compete with the histidine tag that is present on the protein. So what we want to do is to actually see very fast if there are some histidine that can compete. This is the alcohol dehydrogenase that we are using, and as you can see, you have these histidine. There are 2 histidine that can actually we can ask ourselves, they can compete with for our reaction, but here you can see that they are buried inside the structure, so we can suppose that is not so high the competition. But if I'm using the 3D protein imaging to actually see if, while talking with my boss Valeria, we can see that there is some histidine tag, some histidine on the surface of this protein. Maybe we are talking about changing protein. It's a fast way to see, okay, we can use it or not, or it can be used also for other protein for other scopes. And this is the website. You can see the consortium here of the Enzyme project. There is here the Enzyme link if you want to know more about this project. And if you want to try the protein imaging, you just have to go to the 3dproteinimaging.com, and you will have the viewer easily accessible. You can use also in classes. So for your student, for now it's free, so you can actually use to teach to your student how to work with protein. And you can follow us on the social media. And if you have some doubts or you want to suggest us something, you can write us to our email. And also we have published a paper. So if you want to use our work in one of your paper or your thesis, remember to cite us. And also we are promoting ourselves actually, and if you need a cover for a paper or you need a more complex figure, you can ask us also by sending me an email or by writing to the assistants. And if you have some question, I can answer to you in Spanish, English, or Italian.